So I'm going to do a number of things and bounce around here. I'm happy to take questions. I can't see if questions come up, so Tom, pause me.
Yeah, I'll keep an eye on the chat box if anyone has a question.
So please ask questions. This is very much for benefit, and I may end up not where you're wanting this to be, but happy to go there. We use facts every day in consulting. We do simulation every day within our consulting. It's sort of how we do this. It's how we function. And so happy to share positives, negatives, good practices, life lessons in all of this. I love this picture. This is actually in Tom's home country. Somebody sent me this picture of a Simbulance, a mobile simulation vehicle, and I thought this was so cool. I want one of these, and I want it, and of course it would have facts on board. If somebody needs a clinical trial designed very quickly, a good clinical trial, let's send the Simbulance and let's create a good trial design very quickly. Unfortunately, I don't think that's what this does, but boy, that would be neat. There are things we're involved in pandemic scenarios with BARDA, where there are emerging aspects where they want clinical trials designed very, very quickly. So this does happen, and we're involved in some of these, and FACS plays an incredibly important role in that, and I'll discuss more of that. Okay, so I'm going to take a weird tangent here. I was asked a while back to give a talk on statistical leadership, and it was a bit of a struggle to think about what is statistical leadership, what are good practices, what does this mean? And so I grabbed some slides because I think it's a good introduction to the role of facts, stat, / simulation in clinical trials, in consulting. And by consulting, I mean the larger thing. Many of you, I'm sure, are working within teams at a pharmaceutical company, and that's absolutely part of this teamwork. So I gave an example, and this is a real example, where we got involved in a clinical trial design, and we started to simulate the design. And naturally, we compared it to a fixed trial design. And the fixed trial was 80% powered for a nice effect size. We'll call that delta. And that trial's ready to go. And of course, it's a quality trial because it's 80% powered for that. We started to simulate the trial design, showing example trials and showing what happened under the null. And then we started to simulate trials assuming delta was true, and showed them that 20% of trials fail when delta was true. And they were disappointed by that. They were quite disappointed by that. Wow, that's a bad trial design. If this drug really has this great effect of delta, and 20% of my trials fail. They were shocked and disappointed by that trial design. Now, I suspect many of you are statisticians listening to this, and you're chuckling at this, and you may be chuckling from your own experiences of that, and we recognize this as statisticians, that that's what 80% powered means. It means you fail 20% of the time with that effect size at delta. But that was not what this was perceived to be by the people creating the design. 80% power to them didn't mean that it's successful 80% of the time under the effect size of delta. It was a statistical hurdle on how you calculate a sample size, and it meant that that was the constraint that I needed. But in no way was that a decision theoretic thing for them. The 80% meant nothing to them in terms of 80%. It was, oh, this is what the statistician tells me the appropriate sample size is. But no way did this measure risk or anything. The 80% was irrelevant, other than that was how you construct the sample set. That's bad. It's bad in many ways, and in many ways, it's our fault. And so I'm gonna get to that. So the fact that they don't understand power as risk, it's just a restriction that statisticians place on a trial design, is bad that they don't understand what power means. And we understand it. We think about things in a simulation way. We think about power in terms of simulation. Simulating trials under the sample space of the design. It's our language, it's what we do. The reason it's our fault is that we make them talk about our language. We make them talk about power, and we make them translate clinical drug development things in our language. And quite frankly, they're not good at it. We know many examples of that in the situation. And the reason it's our fault is we need to speak in their language, and we do the translation. We are the experts in clinical trial science. We understand this part, but yet we make them talk about alpha and power and all of these things that they're not very good at. And that's what my examples of leadership were. So we have this part of it that much of what goes on in the language of this is pure statistics. Power, for example, 80% power. They're saying to us, oh, I want 80% power, I want 90% power, I want a type 1 error of 0.05, I want randomization of this, or I want to do these things. And really, we should be doing this in their language under the setting. So within the leadership, I talked about a great deal of this is we love to talk about the details of our statistics. We can argue endlessly about whether it should be x given theta or theta given x. Of course, the right answer is theta given x, but that's not what this talk is. That's an example of us doing this when outside of statistician, almost nobody cares. It's our science, it's what we do, and all too often we make them talk in our language. If you're discussing with a clinician x given theta or theta given x, we've lost within that setting. So this was what I was describing in this talk about leadership. And I'm going to get to the point of, I'm going to get to how this relates to this whole topic about teamwork, consulting, and facts. So we have to speak in their language. The disease, the science, the drug, the team, the sponsor, drug development, success, the risks of the trial, all of those things, we're better at speaking in their language, and we need to do that. That's leadership. Rather than forcing them to speak in our language, which they're not very good at. And I gave you one example of that, and we all know many, many examples. We can sit there and chuckle, or we can speak in their language and not force them to speak statistics. So much of this goes on, and they say things like, what's the sample size at 80% power? When there's much bigger questions here. What happens after my phase 2 trial? What is success? What's a good no-go decision for the company? How do we incorporate safety and efficacy in this? You'll hear many times we need to understand the dose-response curve. What does that mean? What does understanding the dose-response curve mean? It doesn't mean we need 80 patients per dose and we can do a pairwise comparison. It's almost never what it means is a p-value of 0.05. So can we really understand all of this? The problem was, historically, bridging this gap was really hard for us because answering hard questions like that was nearly impossible with the tools we had. But if we could do that, we would be better statistical leaders, we'd be better scientific leaders in that setting. So, again, I'm repeating myself now, but to bridge the gap, we need to do that. It's our fault that they do these examples that we do. We need to do the translating. The problem is it's really hard. And if you have to rely on analytical calculation, analytical power calculations, it's how we end up with phase 2 trials that are 3 doses and 80 patients per arm. It's because that's a thing we can calculate. And we force everything into that. Now, we have tools that we can answer questions in their language. We have simulations, we have facts. We have modeling within that, longitudinal model, dose-response modeling. We can borrow data, we can incorporate that. We can do adaptations in our trial designs that better answer questions, the harder questions that they really have. We have much more powerful graphical tools to communicate the ramifications of drug development to the difficulties of the trial design. These are all things we didn't have 10 years ago, or it was really, really hard 10 years ago to do this. Okay, so let's bring this back to how this leadership discussions that I gave is back to this point of consulting. Simulation allows us to speak the language of the clinician, the trialist, the sponsor, and not just statistical language. It allows us to make that translation to what's relevant in the trial. We can make exact calculations that are relevant to the important decisions in ways that we never could just 10 years ago. We can do questions about timing of approval. Can we select the right dose in a phase 2 trial? It was incredibly hard to do that before. Does response adaptive randomization improve the probability of getting the right dose? Improve the go, no-go decision-making for drug developers? We can characterize really interesting go, no-go drug decisions that are based on multiple endpoints and utility functions and calculate the likelihood we make the right decision. We couldn't do that without simulation. It's too hard analytically to make those calculations. We can calculate in a CRM the average number of patients treated at a dose above the MTD. Couldn't do that without simulation. It's too difficult. In a basket trial, does borrowing help make the decision on individual parts of the basket? Or does it hurt? Hard to do that without simulation. Timing is really, really hard. The effect of accrual and the optimal accrual rate to optimize drug development decisions. Can't do that without simulation. Enrolling slower improves the ability to get the right dose, but it takes longer. What's the optimal accrual rate? Again, we can answer these very, very quickly now with simulation. Allows us as the science gatekeepers of clinical trials to now do this in consulting, in teamwork, that you couldn't do before. Okay, so simulation is hard. It's really hard. Asked to do those questions, if you have to write code to do this from scratch, it's slow. Years ago in doing a complicated simulation, the Aspen trial was 16 dose response adaptive randomization Pfizer trial in the late 90s. It was 18 months to do the simulation. Lilly's Award 5 trial, which was 4-endpoint response adaptive randomization phase 2/3 seamless trial, was 9 months of unique code. You make errors. You have to validate the code. You can't write simulations without making mistakes in the code. It's plagued by that, getting it right. It's slow in a way that we can't do it effectively. Now, that's getting better. There's code out there. You can do this faster. If you do this for a living, you can do it faster. But drug development moves faster than that. It's only unique scenarios that are slow enough in timing that we could do it. So FACS allows you to do simulations very quickly, correctly, and very close to what's relevant for the team. And so that's the huge thing. So tying this all together, we can answer more important questions in the language of what's important correctly and fast is the value of FACS in this setting. And it changes statisticians' role in all of this. In all of drug development, which is a very positive thing, not just for statisticians, but for drug development. We can help do things better, and now this provides tools that we didn't have before. This is becoming a commonplace, complex innovative designs. This draft guidance was out within the last week from the FDA. This is in addition to the pilot program, if you haven't seen this. So this is now a thing at the FDA. And a complex design is essentially defined by you have to do simulation. Either the design or the analysis is too complex to have an analytical solution. We understand it through simulation. That's a complex trial, and so how do we deal with this? It's a recognition that this is there, this is a positive thing. And so FACS can play a big role in that. Simulation, of course, this is defined by that. Okay, so warning number one is that clinical trial simulation, as you're working with teams in your consulting, means different things to different people. And people are skeptical of it. We know it differently as statisticians, and I'm referring to we as statisticians. In this setting, we will think of it very differently. And if you start throwing around, oh, we'll simulate it, people are going to be skeptical of that. So why do I say that? When people hear about computer simulation that are non-statisticians, they think of it in ways like this. Computer simulation of Rocky Marciano fighting Muhammad Ali. They were 40 years different eras. They never boxed together. But simulations can predict what would happen if they boxed. Of course, a ton of assumptions go into that. Or simulating the upcoming NFL season, who's going to win it all. Predicting. Simulating the track of a hurricane. Predicting where it's going to go. There's some distrust in this. But we're inundated by simulations being predictions in that. So everybody but statisticians, and now I'm lumping things, and I realize that for a purpose, is being predictions. We use it very differently in clinical trial design. If you use FACS, you use it differently. Now you can use it for predictions, but that's a different topic. PK/PD scientists do clinical trial simulations to predict what will happen when it goes in the body. So they even think about it differently. Your management will think of it as you're forecasting what's going to happen in the trial. We use it for what I'll refer to as in silico design. We take a design, we simulate it to calculate the operating characteristics exactly. Clinical trial simulation is exact to whatever level of exact you need. For us, it's numerical integration. It's nothing more than that. But that's not the way it's perceived by everybody else. by the general public, by scientists. So when you say clinical trial simulation, you're essentially calculating the characteristics of a design. You're making calculations where when you say, oh, I'll simulate it, don't be surprised when that's perceived very differently. So we spend a lot of effort explaining what trial simulation is. My warning number 2 when you do this in consulting, it's clinical trial simulation is not necessarily useful as the final task. Yes, there's a role for it there, but it's a minor role. But this is, in many situations, statisticians are going to think of it like this way. The FDA might think of it this way, but the role of facts is so very different than this. So let me explain what I mean by this. You don't go out and design a trial, say, oh, we're going to do this adaptive design, we're going to do an interims at this time point, and, oh, you statistician, go simulate it and put it in the protocol. Yeah, there's a role for that, but it's not the real value here. And why is that? Because almost surely that design is not a very good one, depending on its complexity. If it has any complexity at all, don't be surprised when its behavior is not very good. But you don't know its behavior is not very good in most settings unless you compare it to something else. So the value of this is simulating the design and many alternatives, different sample size, adaptations, thresholds, and optimizing the design. So there's a whole space here of exploration, and that's what FACS is really good at. As a final verification of type 1 error, yeah, there's a role there. In fact, it's actually really good at that, but it's a much more minor role as the value of FACS. So in your consulting, this is only the finalization step. Most of it's going to be the exploration step, and that's what FACS is good at. And I'll show some examples of that. My warning number 3 in this, and this is a warning not to the team you're working with, but to other statisticians. There's a group of behavior that brings a meta-analysis feel to simulation, where you pre-plan all the simulations you're going to do. There's almost a protocol for what simulations you do. This I find incredibly limiting, and I think it's a different purpose brought to this in good meaning, but it's bad. It defeats the whole purpose of exploration. When you start to simulate trials, you have no idea where it's going to go. Now, as you get to do this more and more often, you may have a guess on where this is going to go, but you can't pre-plan all you're going to simulate, and it's very limiting. So this is done in some settings. Let's write a protocol for the simulations we're going to do. We'll have a one-day meeting and share the results, and we're going to know the answer. This has to be iterative to get the right answer. You can't pre-plan everything because where you go depends on the results of those simulations, and almost always you have to iterate. So what does this process look like in using FACS? And this is we write proposals for what we're going to do with teams. How's Barry going to do in this? And this is what this process looks like. Is we create a set of skeleton designs. And that might be what's done in this protocol for simulations is, ooh, let's look at these designs. Then you go off and you do the simulations, and this is where FACS plays a role. And don't worry about getting that first blue box perfect. It's better to do something close in that setting, and because you don't know where you're going to go, you don't want to go write custom code, spend a month, come back, and that first roll of simulations, you change the endpoint and everything goes awry, and then you spend another month on custom code. get something close. If you have an ordinal endpoint, dichotomize it for the first round of the simulation. Create a continuous endpoint as an approximation to that. Do something that moves the ball forward to make sure you understand the big questions and you're getting close. But do that first round of simulations with facts. You do a presentation of them, you share behaviors and operating characteristics. Invariably, that leads to whole new designs that weren't a part of the original skeleton. It might be as simple as different thresholds, different quantities of interest, different posterior calculations. It might be bringing in other data, but it might be adding arms, changing the number of arms in the trial, seamless phase 2-3 where it used to be a phase 2 trial. You do new simulations, new results in that, new designs. This is the process that has to happen for good design, and you can't pre-plan that. You can't, at this stage, know where that's going to go, and I think it's very limiting in that setting. So don't be too prescriptive over here on the left of what you're going to do. Let the results take you where you're going to go with the team. And tying it back into what I said about leadership is that this is the hardest part, is sharing these results with the team and getting the operating characteristics right. And what do I mean by that is it's not just power. And as you do this more and more with facts, you find that in many settings in the phase 2 space, power's really not something that they're terribly interested in. It really isn't. It turns out to be many other things that they're interested in. So when you present this first round of simulations, that's the part of leadership where you need to capture the relevant medical, clinical, sponsor-related things to present to them in a way they understand. This is where you do it in their language, not just power, type 1 error, and things that they're used to asking the statistician for. That also iterates. We usually present things in simulations. The second round, we're presenting all new operating characteristics because we learned in the first round that that wasn't really the relevant stuff. That's this whole wonderful process driven by statisticians, driven by facts, driven by simulations that lead to better design. Through this process, you get a final design. Now, that final design is much more like the meta-analysis planned stuff that happens in the presentation. We take the final design. By the way, back here, we're not validating code. We may be taking shortcuts on the endpoint to get something very close. And there's an art to that. But then we want to get it all exactly correct, validated code, a set of simulations. We archive those results. We verify we're getting the right things from these results into our adaptive design report. That goes into the protocol. So that's the process of facts. And of course, this goes to regulatory. The adaptive design goes to regulatory. This whole thing back here doesn't necessarily go to regulatory in that setting. So that's the process of teamwork and consulting driven by facts. And too often, people think it's just this part. The pilot program at the FDA focuses on this part, and there's reasons for that. They need to understand how to look at a single design that's been proposed by a sponsor and the code for it, the final results. Do we validate type 1 error? The CID program is generally phase 3. That focuses on that, but facts' role plays such an important part back here. Okay, I'll give you some history into facts and how did we get here. Tom will remember there were other people in the room. Mike Krams was in the room. Vlad Draglin was in the room. Tom Park was in the room in about 2006, when we were deciding, how do you build a software tool? And there were different camps when we were building a tool. This was a predecessor to FACS, actually, where there were some that said, oh, let's take custom code of a single design, validate it, and put it into the software. And we referred to this as an aircraft carrier. So it would have lots of different designs that had been used, and somebody in the future can come back and use Vlad Dragolin design number 7. And it's sitting there on this aircraft carrier. And as we do more and more innovative trials, we put them on this platform and people can go to them. And there are packages that are like this. The other possibility is, let's build something that has flexible choices that can be crossed and explored. This may be easier, it may be harder, and that's the tool that then somebody can go in and explore things that have never been done together in a single trial. It's very different than the aircraft carrier. We refer to it as the nucleus in that setting, and be explored. And that was the decision that was made in FACS. And that ties to that whole description of doing consulting and simulation and how FACS was created. So let me pause here, Tom. Tom, any comments or questions from participants?
No, Scott, we've not had any questions come in so
Okay.
far. If anyone wants to ask them, do find the chat box, type in there, and I'll pause Scott and relate a question to it.
So this is an example that happened this month, and I've taken any details out of this. But this is an example of slides we presented of a simulation, the one on the left. This is for the truth behind this, and so I've pulled that part out to make this confidential as to what the drug scenario here was. But these are kinds of things we present to make this very understandable about the design. At a particular time point, the trial means you file with the FDA, you hit a level of success. And even things like putting the FDA flag there and not saying success. Success is a weird term, and it can be misconstrued. But we do things like this. Lindsay's actually online. She made these slides. Was easier for them to understand. Or stop is futility. Futility is a weird thing. It's a weird word that can be misconstrued. But we put this to explain it. This was presented to us, ooh, we'd like to do this design. Is this a good design? And under their target scenario, the design they presented, which was doing a test with a larger alpha, if their true delta was there, 24% of the time they didn't get out of their proof of concept phase 2 trial. Their power was reduced to 75% when they thought it was over 90% if they did that as their trial design. We iterated it, we created a predictive probability rule, and all this was done, and that went down to 1.4% rather than 24%. Yeah, under the null we showed them, and it had still very good behavior in that. Originally, this project was one where they were skeptical. And I feel like the team was somewhat told by upper management, you should explore innovative simulation and designs. I think they were a bit skeptical. When we started to simulate this and present it in their scenario and all of that, this progressed to not 1 or 2 looks, it progressed to 5 looks, exploring randomization ratios. Can we change it during the trial from phase 2 to phase 3? A number of aspects to this became a much more complicated design, and they trusted the simulations after they were initially skeptical of it. I don't know what this means. I've never done this before. And they were able to trust it as making calculations and comparing different designs on the language that was important to them. So that was an example. Here was another example this month where it's a phase 3 trial, 2 active doses. They want to do 1-to-1-to-1 randomization versus a placebo, slow enrollment, rare disease, about 50 per arm was the design they wanted. And it was, we don't really know if this is the right trial design. Let's call the berries. 4-week endpoint, so relatively quick endpoint. And when the discussion came, well, maybe those 2 doses, given the slow enrollment, the reasonably rareness of this, do you need 2 doses? During the trial, could you have decisions and drop one of the arms, which could create the ability to finish much sooner if your drug's effective? You could stop the trial for futility, maybe a flexible sample size. Why is 50 the right answer? And essentially, we were told, no, the trial is 80% powered at 150 patients. You're not going to know the answer before that. You can't make good decisions on dropping arms because 80% powered is 150. You don't know the answer till 150. It was this, and again, that goes all the way back to the beginning, where they don't understand power, all of these things, misinterpretations. It's our fault that they say things like this. And it was, we had to back them away from this and say, well, they're trying to make quantitative things by non-quantitative people, and that's what we need to do with facts. Now, if I said to them, I'll come back in 2 months and show you whether this works, they'd have said, oh, no, we're going to go on. This is a good design. We're done. But because we can get back to them in a week or 2 with relevant questions about can you do this well and how well, I know this project from doing this for a number of years is they're going to add it in there. It's going to turn into flexible sample size. I don't know what the right thresholds are. I don't know what the right range of sample size is. But I know it's a better design than this single fixed design. Because we can do it quickly, FACS is going to allow us to get it there. Okay. Here's an example of power of simulation that you can't really do any other way. These slides, you'll see 2014 on here. These been approved by the pharmaceutical company Ferring for public disclosure. By the way, these were actually presented, these slides were presented at an EMA conference on designing better phase 2 trials. Regulators, I think the EMA, well, not just I think, it was the goal of this is that they didn't think the phase 2 trials being run were getting great answers. Can we run better phase 2 trials? And this was a trial that went through the EMA, and they asked that this be presented at that conference. It was a phase 2-3 seamless trial, and rather than a traditional, this is sepsis, rather than a traditional trial of 200 patients and make a decision, it could range from 200 to 800 and try to get the right answer. Now, the negative of that is 800 is a big phase 2 trial. So we did a seamless phase 2-3 trial. That if it gets that big, that data is included in phase 3, and the trial expands to a traditional-looking phase 3. But we don't lose those 800 patients. They're included in the primary analysis at the end of phase 3. So from regulatory perspective, this is more likely to get the right answer in phase 2 about the right dose, about the go-no-go decision, than a simple little phase 2 200-patient trial. That's what you do if you can't include the data in phase 3 because it's too expensive to do 800. So that's the general structure of this trial. We spent a lot of time with Ferring on the question of go-no-go. how do we make the go, no-go decision at 800 patients? And it's not about power necessarily. Power plays a role, but it's a development decision. So what we did is we presented the following plots. And why is this something you can't do in other ways? Because we can look at what would have happened if you didn't stop. So right here at that red point, or during the 200 to 800, you could say go to phase 3. And we wanted to create a rule for that. So large investment, they increase the number of sites, and they go to 1,800 patients from this earlier part in the trial. So we presented that on the left, we simulated, there's increasing efficacy from A to E. Of course, E is more effective. 0 is the null. This is the that what shows on the left is the final result if you go to 1,800. So you always go. And there are 2 endpoints in the trial, mortality and morbidity. Everything to the right and up is good. Everything down and left is bad. The final result of the trial for statistical significance is green for positive, black is failed. Now, the question is, can we, in phase 2, stop black trials and succeed with green ones? But it wasn't just black and green, it was the clinical relevance of the green. There are certain values in here that they weren't terribly interested and not sure that it would be used a lot. Of course, things that move up and to the right are clinically beneficial. So there's a dance here with clinical and statistical significance in this range, and they wanted to also understand the role of the go, no-go on those, which power doesn't necessarily get you. And that's partly what I mean by be careful saying success. Success means different things to different people, and power is such a blunt instrument. So what we did is we said, okay, if the predictive probability threshold, and by the way, everything on the left is 1,000 trials from each of the 6 scenarios. So there's 6,000 finished trials to the left, and it shows the power in green and failures in black. If we did predictive probability of 1% of winning phase 3, that you would stop and not go, we turn the final result. You're doing this at the go, no-go in phase 2. That red line, you're making that decision. And we can go back to the finished trials and say, what would we have done earlier? And the pink ones are the ones we kill if we do 1%. And the ramifications to power, which are not irrelevant, are shown to the right. So under the null, about 20% of the time, we would kill a trial that would go on to be a failure. That's a good thing. We would rarely change a green result to failure. That's something they're worried about. They'll turn bright red on the right. But you can see we're killing the worst of the 1,800 results early with a 1% rule. Then we say, okay, what about 5%? Notice some of these trials that would have been successful turn bright red. Several of them out of the 6,000 actually are clinically important. These, while successful, aren't a big deal. And then we want to turn these black ones to pink, and you can see what happens under the null. And C is their scenario of particular interest. We lose about 0.5% of trials if we use 5%. If we do 10%, if we do 15%, if we do, sorry, I cut this off. Sorry. here we do same thing, 25%. I've locked in blue. Blue was the original futility rule during phase 2, and then at that final seminal point of go, no go at 800, you had to make a decision. Is this rule that's the pink here? So we looked at that of 25%, 30%, and so on. And what was interesting is when they shared this with the company, they didn't tell them the number 50% or whatever. They blacked that part out at the top out, and they showed them the graph on the left and the graphs on the right. And then the executives went through, and when they looked at this, they said, oh no, that's way too aggressive. In truth, if C is right, we're killing our power by 15%, and we're losing very marketing and clinically relevant results when we do that. And so we don't like that, and they walked through this and picked this one right here. This was the balance, and the whole process of this was very understandable, and they loved it. So this was something you can't do without simulation in walking through this from the particular standpoint. It's the unique ability to carry a trial to the end in the counterfactual that it would continue and be able to show the ramifications in their language in drug development. And it turns out 25% became locked in the protocol to make the decision in the trial. From that. And by the way, initially they wanted 80% as that, because 80% power. I want 80% power. And the understanding of the quantity predictive probability of winning phase 3 to power was something that if we said, you tell us the predictive probability, that goes all the way back to the leadership. I think it's the wrong. We're asking them to do something that is hard for them to do. It's not understandable. It's in our language. Predictive probability is our language, even though it's an incredibly relevant thing. In given time, you can explain predictive probability much better than conditional power or other things. It really was still not what we needed to do. We needed to speak in their language, and this was much more in their language. And they didn't even have to pick a number. Through this, they integrated over this and made a decision. So my other warnings of this, be careful of the terminology and facts in your presentations. A lot of that is fact-specific terminology that the more time you spend there, the more it makes sense and it's nice, but terms like flip-flops, inconclusive, success, the word success and futility in there, if you put that on a table, it can be misconstrued. So a lot of the things that are in fact, you want to redo in your own graphs, put it in the terminology of the clinician of the trial, the go, no go, we submit to FDA, we stop development. Those are much better than inconclusive, success, those types of things. The presentation of the results are so incredibly important. And this is something you hear over and over again. But the same result presented in a dry table with bad labels is going to lead to the wrong decision on the trial design that wouldn't be had if you did detailed simulation, detailed presentation in their language in ways that are easy to see and easy to understand. One hint I try to do when I'm making a presentation is, if it were up to me and it was my decision, what would I do? And a lot of times, well, okay, I would compare this on page 7 to this on page 11. I'd pick this, and I realize my slides aren't very well set up to make that decision. And how bad is that I'm presenting slides to somebody who needs to pick a design, and I can't even make the decision I would make with what I've presented to them. and I usually restructure it and present and compare things in different ways. And that usually leads to a much better presentation. Example trials are critical. Again, operating characteristics, averages are hard to understand. What do the data look like that lead to a decision where I would stop? And present that and show that to them. Bring them along that this is not a black box, that this is them making the decision. I understand what the data looks like. The algorithm's my friend and not a black box. Show the data, the conclusions, and all of that, and this is all going to go much easier and create a better design. So I've got a whole set of slides on IceCap. By the way, if you've seen me present IceCap before, this has now been funded by the NIH. So there's 10 different durations. The design was created through this whole process, and so. But I don't have time for that, and it's the same things I've been saying. So I will stop here. And questions, comments, Tom?
Still had no questions come in, Scott. I mean, personally, I just want to grab my laptop and rush out and consult to somebody. That was a terrific presentation. I hope everybody else gets the same glow of energy and as I did of it. Yeah, thanks so much. I think actually this was such a incredibly useful presentation. We should look at putting a link to it on our website.